All right, let's dive into the absolute heart of what makes life, well, life. We're talking about proteins. These are the master molecules, the tiny engines and the literal scaffolding inside every single living thing. So let's get into how these incredible little machines actually work.
So just take a look at these two for a second. On one hand, you've got the raw power of a rhino's horn, and on the other, the super delicate, almost magical light of a firefly. I mean, what on earth could these two possibly have in common? Believe it or not, the answer is proteins.
And that right there, that's the big puzzle we're going to tackle. How can one single class of molecules build things that are so ridiculously different? It seems totally impossible, right? But the secret? Oh, it's surprisingly simple and elegant.
To get started, let's just look at the name itself. Way back in 1838, a chemist named J.J. Berzelius came up with the word protein. It comes from the Greek proteos, which means primary or of the first rank. He just knew, even way back then, that this stuff was the main event. And wow, is he right.
So how does this primary substance pull off so much variety? Well, it all boils down to this one number, 20. That's it. Every single protein in your body, in that rhino, in that firefly, in every living thing on this planet, is built from a standard toolkit of just 20 building blocks.
And we call these building blocks amino acids. Now here's the big idea, the key to this whole explainer. Think of protein structure as a language. If it's a language, then these 20 amino acids are its alphabet. By arranging these 20 little letters in different ways, life can literally write the instructions for anything, from a muscle fiber to a spider's web. So, if these are our letters, what do they actually look like? Let's zoom in a bit and look at their basic anatomy, because that's where you start to see how they all work together. You see, this common structure is the key. Almost all 20 of our letters share this exact same format. You've got a central carbon atom called the alpha carbon, and attached to it are two groups, an amino group and a carboxyl group. This backbone is basically identical for almost every letter in our biological alphabet.
So you're probably asking, if the backbone is the same, what makes them different? Well, it's this piece right here, the side chain, which we call the R group. This is the part that changes. The size, the shape, the chemical vibe of this one group is what gives each of the 20 amino acids its own unique identity, its own personality.
Okay, but before we start building words with these letters, there's one more really fascinating and honestly kind of mysterious rule about their structure we need to talk about. It's this fundamental choice that all of life on Earth made a very, very long time ago.
So that central carbon atom? In 19 of the 20 amino acids, it's attached to 4 completely different things. In chemistry, that creates what's called a chiral center. And what that means is the molecule has a handedness, which allows it to exist in 2 different mirror image forms.
And the best way to get this is to just look at your own hands, right? They're mirror images of each other, but you can't place one perfectly on top of the other. They're non-superimposable. Well, amino acids are exactly the same. They come in a left-handed L form and a right-handed D form. Chemically, they're the same stuff, but in 3D space, they're different.
Now here is the mind-blowing part. Life had a choice between 2 completely different mirror image alphabets. And for reasons that probably have to do with stability and just keeping things consistent, it went all in on the left-handed L version. The proteins in you, me, and pretty much everything else you see are built with L amino acids.
All right, so we've got our 20 left-handed letters. Now, to understand how they form words and sentences, in other words, working proteins, we need to see how they're grouped. Biochemists sort them into families based on their chemical personalities.
Remember those unique R groups we talked about? Well, their most important trait is their polarity, which is really just a fancy way of asking, do they love water or do they hate it? This one simple preference pretty much dictates how an entire protein chain is going to fold itself into a working machine. So let's meet our first family. We can call them the water-fearers. Now, the scientific name is nonpolar or hydrophobic, but water-fearers I think really paints the perfect picture of what they do. This group includes guys like glycine, alanine, valine, leucine, isoleucine, proline, and methionine. You can think of them as the shy introverts at the molecular party. So, because their R groups are nonpolar, they absolutely hate being around water. Imagine you drop a long chain of these amino acids into the watery soup of a cell.
What happens? All these water-fearing bits frantically try to get away from the water. They all scrunch up and hide together on the inside of the molecule, and that powerful drive to hide is one of the main forces that makes a protein fold into its unique 3D shape.
And that's just one group. Of course, there are other families too. You've got the aromatics, you have the polar or water-loving ones, and then you have ones that carry positive or negative electrical charges. Each family brings a different tool to the table, giving life a full chemical toolkit to build with.
Okay, let's put all the pieces together. We have our alphabet of 20 letters. We've sorted them into families with different personalities. So how do we get from that to all the action we see inside a living cell?
Well, it's basically a four-step process. First, our DNA spells out a very specific sequence of our 20 letters. Second, that long chain just spontaneously folds up, all driven by the personalities of its R groups, like our water-fearers diving for the center. Third, this folding creates a stable, unique 3D shape. And finally, that very specific shape is what allows the protein to do its very specific job.
And that brings us all the way back to our rhino horn and our firefly. The difference between them isn't the letters themselves, it's the sequence, the order, and the length of the words. The keratin protein in the horn is like a long, repetitive, structural sentence, but the luciferase in the firefly is a compact, intricate little sentence that's designed to perform a chemical reaction. It's the same alphabet, just writing completely different stories.
So, when you think about it, the big takeaway is this. The mind-boggling complexity of life isn't built from complicated ingredients. It's built from the nearly infinite combinations of a few very simple ones. And that just leaves you with this one last thought. If all the life we see around us is just the stories that have been written so far, what incredible new stories, new functions, and new structures are still out there, just waiting for us to learn how to read them from this simple 20-letter alphabet?